So it is my pleasure to introduce Christian Steinebach. He's a postdoc at the University of Bonn's Pharmaceutical Institute, where he focused on the synthesis of PROTAC molecules. He successfully finished his PhD in pharmaceutical chemistry at the University of Bonn in 2020 and has since aspired for an independent academic career with a focus on induced proximity modalities. Alongside many internationally renowned scientists, he was able to identify selective PROTAC degraders of E3 ligases, cereblon and IAPs, in addition to homo and heterobifunctional ubiquitin ligase degraders. His team developed PROTACs that target pharmaceutically relevant kinases, epigenetic reader proteins, and DUBs, and his mid-chem portfolio includes the discovery of optimized E3 ligase ligands, as well as the improvement of physicochemical features of PROTACs. I'm very pleased to hand it over to Christian.
Thank you very much, Catherine, for the kind introduction, and welcome to my talk. So in today's talk, I want to present different strategies to rewire the Ubiquitin-Proteasome-System. We have three pillars in the lab of TPD research that consists of, on the one hand, hijacking the UPS in a classical PROTAC and molecular glue approach to degrade targets in cancer. But we have also generated examples where we targeted specific members of the UPS, such as E3 ligases or DUBs. And last but not least, as a medicinal chemist, we do also development of new E3 ligase ligands. Over the last years, we have generated several case studies for such applications. For instance, we provided manuscripts on CDK6 degraders, BRAF, HDAC6. But in my today's talk, I want to focus on PROTACs that target the ligase cereblon, IAPs, and the DUB USP7. And in the second part of my talk, I want to present two case studies where we developed new ligase ligands for cereblon and VHL. Before I come to the main part of my presentation, I want to briefly mention how we jumped into the TPD game. So my mentor, Michael, he has a long-lasting track record on thalidomide research, in particular with respect to the synthesis of thalidomide analogs that were previously investigated as inhibitors of angiogenesis long time before the rise of the TPD modalities. For instance, this compound here, CPS45, is a very strong inhibitor of angiogenesis, as you can see in the microscopy image. In the year 2015, I joined Michael's lab as a PhD student, and we developed some more angiogenesis inhibitors. But you are also well aware that at that time, there were some breakthrough discoveries with respect to the identification of thalidomide's primary target, and then also hijacking cereblon as an E3 ligase that was published at that time. And we followed that, of course, with great interest. And we had a brief look around who is working in the field of cereblon biology and TPD in general. And we were really lucky to meet Jan Krönke. We invited him to Bonn to give a talk on his research. Jan Krönke is a well-recognized scientist working on TPD. And most importantly, during his postdoc in the US, he worked on elucidating the exact mode of action of IMiDs like pomalidomide, which is a well-known molecular glue example that leads to the recruitment of IKZF1 and leading to its degradation. So I remember sitting in the office with Jan, and we were brainstorming what we could do with respect to TPD modalities, and we were discussing this great paper by Georg Winter on novel BRD4 degraders. And after some time, we came up with the idea, let's do a homobifunctional pomalidomide derivative. These compounds later on also coined as homoprotacs induced very efficiently a self-directed ubiquitination of cereblon, leading to its degradation via the proteasome. However, these molecules were insofar self-limiting as the recruited ligase was also degraded. Later on, we developed heterobifunctional molecules that we coined CRBN 655 VHL that refers to this linker pattern where a second E3 ligase was recruited, and very surprisingly, just CRBN was ubiquitinated and degraded, and this compound possessed a nice Dmax and DC50 value. We can also see that in this comparison here, where we compared the homobifunctional PROTAC and the heterobifunctional molecule. The heterobifunctional molecule was much more potent and was also superior with respect to the avoidance of neosubstrate degradation that was only observed here at higher concentrations, possibly due to a hook effect. The selectivity of these both chemical tools were then later on also characterized on the proteome level. But most interestingly, these chemical tools that induce a transient knockdown of cereblon was really well tolerated by all the cell lines that we investigated, indicating that cereblon is dispensable for the cells. And these results were also in alignment with DepMap database data, where cereblon knockout was also well tolerated. At the same time, this raises some concerns with respect to hijacking cereblon for TPD purposes, because it might be highly possible that a genetic inactivation of cereblon might cause IMiD or PROTAC resistance. And so far, it's good to also have other workhorses for TPD purposes. Besides these nice tool compounds to degrade cereblon with a heterobifunctional approach, there are several other examples in the literature. For instance, these two MDM2 degraders were described that are also therapeutically interesting. Recently, also these key Bonn degraders have been published. However, a match between two different E3 ligases that still had to be played was the following one. We recruited VHL or cereblon to different IAP members with a heterobifunctional molecule, and this leads to a pan degradation of the IAP members. In this study, we created 3 different series of compound. We fused the IAP inhibitor via a linker to the VHL inhibitor. We have also explored a different linker exit vector, and in the 3rd series, we recruited cereblon. In all 3 series, we realized the same linker pattern that consists of alkyl and PEC-type linkers. And then we evaluated these compounds with respect to IAP degradation. This data can be seen here. So we evaluated CIAP1, CIAP2, and XIAP, and also the ligase that have been recruited. And to sum up, we can say that most compounds induced a pan degradation of the IAPs. So CIAP1, 2, and XIAP were degraded to more or less good extent. Only in certain cases, also the recruited ligase was degraded, which was not often the case for the cereblon series. However, within this series of compounds, we could also induce a certain type of selectivity by the selection of the linker and the ligase. For instance, this compound slightly selective for CIAP1 and XIAP, whereas, for instance, this compound is slightly selective for XIAP. However, we wanted to explore more this pan-IAP degradation feature, and we have further developed the compounds, and the final hit compound was CST626, which is a very potent pan degraders of all the 3 IAPs with nanomolar DC50 values. The selectivity for the IAP degradation was also confirmed by proteomic analysis. And in this Western blot, you can nicely see again that this heterobifunctional molecule induces a pan degradation of all the 3 IAPs, whereas this was not possible with monovalent or bivalent SMAC mimetics. This also translated into potent cell-killing properties that were at least competitive with advanced compounds such as AZD. While these were the first examples highlighting our work on E3 ligase degraders, I now want to talk about more recent work on targeting deubiquitinases. We believe that targeting DAPs with PROTACs is very exciting because DAPs are overexpressed in several cancers. We can also accelerate the degradation of their respective substrates. that might otherwise not be druggable with small molecule inhibitors. And last but not least, with such a tool compound, we could identify novel DAP interaction partners. DAPs belong to a vast class of proteins in which the USPs represent the major subclass. And here we focused on the DAP USP7. USP7 is well studied in the context of regulating the MDM253 axis. And as mutated P53 is involved in many human cancers, USP7 remains an exciting drug target. However, in the context of AML, we found that when we treated AML cell lines with respective USP7 selective inhibitors, the viability was impacted independent of the P53 status. And this indicates that there might be some other targets of USP7 responsible for these effects. Accordingly, the project's main goal, the project was evaluated by the postdoc Arunima in Jan Krönke's lab. The main goal was to understand the role of USP7 in AML pathogenesis, and Arunima used the combinatorial approach using multiomics, CRISPR-Cas screens, in vivo studies, and also drug combination studies to elucidate the targets of USP7 and their role in AML drug resistance. So it was only a matter of time until Jan phoned us and asked if we can synthesize a USP7 degrader. And here we started from the selective inhibitor XL188 and through the add of different linkers and cereblon recruiting scaffolds, we were able to quickly generate this first-in-class USP7 degrader CST967 that we published last year. It worked pretty well in multiple cell lines that we tested, and also it worked in AML cell lines such as HL60, but also primary AML samples. However, our expectations with respect to cell-killing features were not yet met with this PROTAC, often performing worse than the related inhibitors. Changes that we did in the second generation of USP7 PROTACs were swapping the E3 ligase recruiter to VHL-based compounds. And most importantly, we changed the USP7 inhibitor scaffold, whereas the first generation of PROTACs were based on XL188, which have some disadvantages with respect to target affinity. We later on changed to the so-called compound 5 scaffold. And here it was critical to maintain an H-bond interaction in the exit vector. So if we kept this H-bond interaction, we obtained inhibitors that possessed good inhibitory activity. However, if we changed these structural elements, the inhibitory activity dropped, and it was even worse for rigidified compounds, as here on the bottom. So with these structure-activity relationships and the new E3 ligase VHL, we were able to synthesize significantly improved USP7 PROTACs that outperformed previously described cereblon-based PROTACs. They worked really well in AML cell lines that we tested. And finally, we also got the results with respect to cell-killing features that we wanted to see. The VHL-based PROTAC is also significantly better than the epimer, the inactivated VHL-based PROTAC. And it also outperformed the cereblon-based PROTAC, and in certain cases also the inhibitor. Now, in the second part of my talk, I want to come to recent work on the development of new E3 ligase ligands. I want to present two case studies, one for cereblon and the other for VHL. Jumping back to this slide on angiogenesis inhibitors, we found that the fluorination of the compounds was beneficial for the biological activity. However, we found out later on that these barbituric acid-derived angiogenesis inhibitors did not bind to cereblon. This was only maintained with the aminoglutamide portion. However, we found that the polyfluorination of the benzamide analogs were also performing well with respect to cereblon affinity. You can see here that the non-fluorinated compound has significantly lower IC50 values. We hypothesized that not only the lipophilic nature and the electronegativity of this compound had improved the properties, but we postulated that an intramolecular hydrogen bond interaction might be beneficial for the compounds. And to test this hypothesis, we synthesized a series of related benzamides. For instance, this compound that just has one fluorine here in the ortho position was again a better inhibitor of cereblon compared to the non-fluorinated analog. This trend was also observed for other compounds in which the ortho substituent could be included in such an H-bond interaction. For instance, compound 11D was a decent cereblon binder comparable to pomalidomide. Thanks to the great contributions by Markus Hartmann at the Max Planck Institute in Tübingen, we could not only obtain the binding data, but we could also crystallize all the benzamide type ligands. And here it was confirmed that these ortho substituents do indeed form an intramolecular interaction here with the NH of the amide bond. Besides these very promising in vitro data, we were also very happy to see that the neomorphic features of the benzamide type ligands were not so much pronounced as for classical imide scaffolds. We selected compound 8D with the fluorine substituent in the ortho position and 11D with a methoxy group for further investigations due to the fair binding affinity and also the neo-substrate sparing features, along with, in general, good stability. Of course, we had to meet the expectations of the TPD community to make some nice BRD4 degraders. However, it was not that easy as we expected it to be. The challenge was here to attach the linker to this aromatic moiety, especially in the presence of the fluorine. We tried all kinds of palladium catalyzed transformations, but it did not really work out. So the key to solve this problem was a copper proline catalyzed Ullmann type reaction. So we started from this acid building blocks. We could attach all these Bock-protected, mono-Bock-protected linkers. And from these intermediates, we then assembled the final PROTAC molecule. Compound 43H was then further selected for in-depth characterization, again done in Jan Krönke's lab. We synthesized some analogs of 43H, in which we incorporated the methoxy group that was previously shown to have more potent binding affinity. And indeed, these translated into also more pronounced BRD4 degradation, whereas the non-substituted analog, again, was also inferior with respect to target degradation. The methoxy compound was also evaluated in proteomic analysis, again showing the selectivity for BRD4, 3, and 2. And last but not least, and this was really a great result, the ligands as well as the derived PROTACs were indeed neo-substrate sparing. So here you see the degradation capabilities for ICASET-F3, and this was significantly less pronounced as compared to PROTACs that derived from classical imide scaffolds. We wanted to transfer these desirable features to other targets, and in collaboration with Finn Hansen, also here at the University of Bonn, we synthesized HDAC6 targeting PROTACs. And this was done by the PhD student Felix. Felix employed their previously described HARES approach, in which the PROTAC was synthesized on solid support. And again, these benzamide type PROTACs with a methoxy group or fluorine here in the ortho position were potent degraders of HDAC6 and also slightly better than the reference compound. Finally, we were also really pleased to see that this structural feature was again embedded into Avinnar's most advanced androgen receptor PROTAC ARV766. To sum up this case study, we believe that through our multidimensional approach, we were able to convert moderately active ligands into PROTACs that are competitive with IMiD-based degraders, but they have superior characteristics with respect to the stability and the neo-substrate-sparing features. In the last few minutes, I want to share another great collaborative story on the development of improved VHL ligands that we also published this year together with Alessio Giuli. However, a facile synthetic protocol was needed to synthesize these ligands that have now a new substitution pattern here at this central phenylene moiety. And this actually dates back to some work that I did during my PhD thesis. Indeed, this phenylene moiety was completely underexplored. And here we wanted to explore now hydrophobic interactions within the binding pocket. At the first glance, this looks like a minimal adjustment coming from the inhibitor VH298 to the now most potent inhibitor compound number 30. But in reality, it was a tremendous amount of work that was also done here by these chemists. And I want to guide you through some of these efforts. As mentioned before, we wanted to explore hydrophobic interactions here at this central phenylene moiety. And therefore, we installed different substituents like a methyl group, methoxy, fluorine, or chlorine. We also did that here at a different position. And in another series, we installed 2 of these substituents to really force one of these substituents into the binding pocket. We then evaluated these compounds utilizing a fluorescence polarization assay, and we pretty fast come to some hit compounds that have similar binding affinity as VH298. For instance, this compound with a fluorine or this compound with a methyl group. Next, we wanted to improve these compounds in some further iterations. And we substituted this cyanocapping group with fluorine. And at the benzylic position, we installed an S-configurated methyl group. Both modifications were previously reported to improve the binding affinity. With respect to this double substituted derivatives, this compound with this methyl group, fluorine, methoxy, and fluorine again, was now the best compound. We then postulated that a rigidification of the benzylic position could again improve the binding affinity, also based on docking. However, these were against our expectations, and all these rigidified compounds, you can imagine how difficult these are to synthesize, had very low binding affinity. I will come back to that later on and explain why. Then we jumped again to this compound, initial hit with fair binding affinity, and we applied these rules. So we incorporated fluorine and we incorporated the S-methyl group, and then we came to compound 30 that is so far the best inhibitor tested. My former colleague Lan had the opportunity to visit Alessio's lab for 2 short research stays. And at that point, I want to thank the whole Center of Targeted Protein Degradation team for hosting Lan. During her stays, she could support several crucial experiments that were performed in Alessio's lab. And one of them was also the crystallization of our most promising hit compounds. So here you see the co-crystal structure of compound 30. And interestingly, this methyl group does not point into the binding pocket. However, this methyl group is insofar critical as it pre-organizes a low-energy ligand conformation, and this can then enter a really nice T-shaped orientation for pi-pi interactions, here with this tyrosine 98, as can be seen here. Very optimal T-shape, which is not that optimal in compound 33, and this might explain the differences in binding affinity. And this was not well pronounced here in the case of compound 37, which explains why these compounds are inferior. Compound 30, along with other selected inhibitors, were then also evaluated in cells for their capacity to stabilize HIF-1 alpha. And here you nicely see that we really improved the compound. Compound number 1 is VH298 over the course of these iterations. In general, the in vitro binding affinity correlated well with the HIF-1 alpha stabilization capacities. With that, I also want to end my tour through our recent publications on ligase and DAP degraders, as well as the selected case studies for Cereblon and VHL Ligand Development. Finally, my special thank goes to all the main collaborators, especially those that have contributed to the results presented today. Of course, all co-workers, also in my group, my mentor, Michael, and last but not least, thanks to the funding agencies to support the projects. And thank you for your attention.